So you're me and you're in combined science class, and you're supposed to be learning about creating homogeneous catalysts, which you figure is probably something to do with the top 10s of the feline side of YouTube. But every time you try to pay attention, you find your teacher's explanations of catalysts conjure up nothing but images of kittens stuck in jars and boxes, or battling vacuum cleaners and toilet rolls. You pledge not to come back to another combined science lesson, at least not for a while, and so you begin to think of any excuse you can of how to get out of coming to school. And so you begin having one of those flashback things from season 1 of Daredevil, except instead of going back to a boxing ring, you find yourself in the Middle Ages, which is great. Everything seems nice and happy and simple, but wait! Suddenly, everyone starts dying around you of, it seems, the bubonic plague. You congratulate your subconscious for bringing you back here because you realise this would be the perfect lie to tell to your parents, that you have this horrible, ugly plague that will mean, undoubtedly, you will have to take a few days of school. So, you begin to paint little red boil spots on your face and hands, but then wait, you realise nobody gets bubonic plague anymore, which is weird, because diseases must have been around since time begun, and they're still here now. But then you wonder if in the beginning, those cavemen guys just sometimes dropped dead, and more religious among them just made mournful glances and grunting sounds indicating it's the work of the unknown. But to make sure, you decide to go home and boot up your newly updated Windows 10 and Google the origin of bacteria, and you find, that indeed eras across all the way back to the ancient Greeks have had horrible diseases which have killed them all. You quickly find that the Egyptians too had to deal with a whole load of illnesses, though their cures usually involved getting people to bleed to unblock their channels, or praying. People got sick all the way across to the 14th century of Britain, with the Black Death, and again in 1666 with the Great Plague of London, and finally to more modern times where we've had yellow fever and smallpox epidemic, until now when every other day people talk about AIDS, swine flu, or Ebola. You also find that you were right, the cavemen didn't get sick a whole lot because disease only really began to spread in large numbers when civilizations grouped together. Why? Because, firstly, irrigation began, which caused naturally hot and dry stretches of land to become all moist and good for land, basically a breeding ground. And secondly, the domestication of animals. Disease often finds itself living in creatures, and our frequent contact in this unhygienic environment allowed the spread of disease to multiply exponentially. But wait, this makes it sound like diseases are alive. Appreciating at this point just how deprived your education has been up until now, you head back to your computer to research types of disease causes, or as you quickly find out, pathogens, which are microscopic organisms that spread and multiply inside our bodies or wherever, making us feel ill. These microbes spread either by contact, or in mistreated water, or in the air, replicating quickly in favourable conditions, hence why discovering irrigation or domestication killed a whole load of people. Since then, as noted, pathogens have spread several times across the globe in epidemics, and we fought them, basically with vaccines and inoculations, which, went so far until something changed in 1928. Scottish scientist Alexander Fleming noticed a mould growing on a petri dish which was inhibiting bacteria growth. He called this mould penicillin. Wertheim and Elric had synthesized arsphenamine, which was used to treat syphilis in 1907, but following Fleming's discovery, antibiotics could now be mass-produced and used across the world to treat bacterial infections, effectively halting the spread of pathogen pandemics and saving us from disease. Our hero is, of course, penicillin, from which many antibiotics are derived, and because of it, we are saved forever. Right? But hold on, a question suddenly comes to you as you flick through your doodles, and you ask yourself why it was that when these early civilizations, who had no vaccines or antibiotics or any knowledge of what was causing their illnesses, how did they survive the sudden outbreaks of diseases caused from them coming together in cities or towns? Well, they developed something called resistance, which is what your immune system is based on. Familiar diseases are recognized, logged, and prepared against in case they come back, assuming, of course, they didn't kill you the first time. Well, that means our early city builders are in fact okay. After the first 1,000 or so who died, those that survived were immune to infection, and so resistance built up in their children, eventually making it harder for that specific strand of virus or bacteria to find a host, and so it died out. It became obsolete. Of course, that all sounds great, but it rings a bell with you somewhere. The idea of resistance brings up a thought bubble around your head about the common cold. Don't you get infected by that every year? Well, this happens because the rhinovirus, which has about 99 different known strands which cause colds, stores information in RNA molecules as opposed to DNA, which is a lot less stable. And for this reason, it tends to mutate or replicate an error in its genetic strand once every 1.5 in 100,000 base pairs per infectious cycle. And its genome contains about 90,000 base pairs, so that's about one mutation during each infectious cycle. Each time it mutates, it can go one of three ways. Either rendering the virus defunct and useless from the copied error, and so it dies out, or it becomes not dissimilar enough from the original virus to have any effect whatsoever. The new mutated virus dies out when the host bodies, that's us, our immune system, kill it. Or finally, these group 3 viruses mutate to become a new strand to the originals, and so they are able to continue infecting and live on, at least for a short while before it has to mutate again. So it's the ones resistant to our immune systems each time which survive. And actually, you've killed off all the competition for it, so in fact it begins to flourish. You realize that all the other pathogens are doing the same. The HIV virus, for example, has one of the highest mutation rates known. It seems to be able to mutate to resist drugs in a single day. And for this reason, it's actually impossible to eliminate HIV or undo damage it's done. But then it's not just viruses which can mutate, it's bacteria too. And this has led to what's been called a global crisis of antimicrobial or antibiotic resistance that is set to cost the global economy £64 trillion by 2050 and 300 million premature deaths worldwide. Because even a simple paper cut could potentially become lethal, our antibiotics are quickly becoming useless. Soon we could be facing a very harsh future of diseases to which we have no cures. So why is this happening? Well, there were lots of infection strands of bacteria that were all affecting a whole bunch of people, but then we introduced a whole load of antibiotics, and that killed and stopped the illnesses and made everyone better, just for a short while. Bacteria replicate extremely fast, and some have that mutated DNA that resists antibiotics, so those ones live on and happily occupy all the live warm bodies left behind by their dead forefathers, and then bam, a whole new generation of bacteria immune to our medicine. Well, hang on, you ask. Why can't we just produce more antibiotics? Don't we just need more molds? And to that, the internet replies, that would be great, but unfortunately only 1% of soil microorganisms, which of course is what penicillin is, can be efficiently tested because only this 1% can be grown on a petri dish. And it's for this reason that the rate of new antibiotic discovery is far slower than bacteria mutation. So what's the future for antibiotic research? Well, awareness is being raised. The problem of resistance is indeed now a global affair, so large-scale international efforts that are made are the most effective. Pharmaceuticals are starting to have to cut down on the usage of antibiotics for only when it's absolutely necessary. Otherwise, its effectiveness could soon be lost entirely. Perhaps the future is not so bleak after all. The latest from work collaboratively by US, UK and German researchers have used a device called an iChip, which cultivates bacteria in diluted soil, thus eliminating the need for a petri dish, which has finally led to the isolation of Eleutheria terrae, a strand of gram-negative bacteria which produces an antibiotic called teixobactin that attacks the lipids of the cell walls of bacteria instead of the protein. These lipids do not change in bacteria mutation, which means this group may have found an antibiotic that transcends resistance, and could be the first in an entirely new generation of antibiotics, which the team affectionately call microbial dark matter. Eleutheria terrae is a huge breakthrough in AMR research. It's the first new strand of antibiotic discovered since 1980. However, teixobactin is a long way away from clinical use. It's a long time before it will be put to market, but still the potential there is huge. Antimicrobial is a huge ongoing field of research, and as you finally bookmark the articles you were browsing through and shut down your computer, you wonder if you could become a pioneering researcher in discovering new effective pathogen treatments, and you endeavour, if your combined science class ever happens to cover microbial research, to listen very carefully.